I think we'll start. Well, good morning, everybody. Welcome to the research rounds. I'm sorry I'm walking because I'm going from one meeting to this. It ran a bit late, but it gives me great pleasure to introduce Dr. Barbara Porto. She is an assistant professor at University of Manitoba, a good friend and a fantastic scientist from whom I've learned a lot about viruses and how they affect neutrophil extracellular traps. She will be giving us a brief overview on work associated with the viruses and neutrophils in context of respiratory infections. I hope you'll enjoy the lecture, and we'll have a lot of time to discuss. And I specifically request the trainees to please ask, because Barbara is a wonderful person who takes a lot of time and effort to explain concepts, difficult concepts, in a very simple way for students and for faculty alike. So, Barbara, I would leave the stage to you. I'm very much looking forward to your lecture. Thank you again for taking this request to present for us.
Thank you so much, Manali. Thank you for your kind introduction. I will try to be as good as you mentioned. And thank you for inviting me to be presenting some of the work we do in my lab here for McMaster. So I'll be sharing with you guys today some of the work that I have been doing for some time, like 10 plus years, and some of the recent work we have been doing in my lab. So I'll be talking to you today about some of the mechanisms of respiratory syncytial virus-induced inflammatory cell death. So as Manali just mentioned, I am an assistant professor in the Department of Medical Microbiology and Infectious Diseases at the University of Manitoba, and also a scientist at the Children's Hospital Research Institute of Manitoba. So let me see. Okay. Okay, so I'll be talking to you about respiratory syncytial virus, or RSV. So RSV is an enveloped single-stranded RNA virus whose genome encodes up to 11 proteins. One of these proteins is called RSV fusion or F protein, and this protein is responsible for mediating the fusion between the virus envelope with the host cell membrane. And this protein is also the target of the monoclonal antibodies available to treat severe disease in infants. And also it's the target of the recently developed RSV vaccines. So RSV is the most frequent cause of hospitalization due to acute bronchiolitis and viral pneumonia in children under 2 years of age. But RSV can also affect the elderly, and it can cause severe pneumonia or even worsening symptoms of asthma or COPD. Therefore, understanding how RSV interacts with the host is crucial for us to better understand and characterize these pathogenesis, and also to develop and/or repurpose new therapies against this viral infection. So once infection is established in the lungs, innate immune cells such as alveolar macrophages and neutrophils are the first immune cells to encounter the virus. So neutrophils are rapidly recruited to the lungs, and together with alveolar macrophages, they are able to phagocytose, release large amounts of cytokines, generate reactive oxygen species, degranulate, and produce neutrophil extracellular traps in the case of neutrophils. Therefore, the macrophages and neutrophil response to infection can critically influence the outcomes of virus infections. So in this scenario, recent studies have revealed that innate cell death plays an important role in determining the pathogenesis of pulmonary viral infections. Therefore, a tightly controlled cell death is beneficial to the host. However, an excess of lytic cell death or inflammatory cell death is detrimental to the host, because these modes of inflammatory cell death, such as necroptosis, pyroptosis, and netosis, can be detrimental once, as the plasma membrane ruptures. These cells will release all of their intracellular contents, including damage-associated molecular patterns, which can cause inflammation and worsen tissue inflammation. So in this table, we can see some of the biochemical features of the different lytic cell death modes. So netosis, during netosis, we will see nuclear membrane collapse, the plasma membrane will rupture, and we will see DNA fiber release. So the result of netosis is the formation of neutrophil extracellular traps, and these neutrophil extracellular traps will be coated, or they will express some of neutrophil granular enzymes, such as neutrophil elastase and myeloperoxidase. During necroptosis, we will see cell swelling, the plasma membrane will rupture, and intracellularly, we will see activation of specific proteins, such as receptor interacting protein kinase 1 or RIPK1, RIPK3, and MLKL or mixed lineage kinase-like protein. And once MLKL is activated, this protein will migrate to the plasma membrane, forming pores in the plasma membrane, leading to the release of all of the cell's intracellular contents. During pyroptosis, the will rupture. In the case of pyroptosis, we will see caspase-1 activation and gasdermin D activation. Once gasdermin D is activated, this protein migrates to the plasma membrane, forming pores, and we see a large release of IL-1 beta, which will perpetuate inflammation. So I will start talking to you today about neutrophil extracellular traps, or netosis induced by respiratory syncytial virus. So in the first study that we did in my lab, we wanted to understand how RSV would induce net release from human neutrophils. So we first tested the effect of RSV fusion protein. So we collected neutrophils from healthy donors, and we stimulated neutrophils with different concentrations of RSVF protein or PMA as a positive control. And we can see that RSV fusion protein is able to induce the release of DNA. So we measure cell-free extracellular DNA using a specific DNA dye. And we can see here that RSV fusion protein induces the release of DNA with neutrophil elastase and myeloperoxidase coated in these DNA fibers. And then we were interested in understanding the mechanisms of RSV-induced netosis. And there are two different modes of netosis, the rapid netosis and the classical netosis. So the rapid netosis happens after 10 minutes of neutrophil stimulation, and the classical netosis occurs after 3 hours of stimulation. So we infected human neutrophils and measured DNA release after 10 minutes or after 3 hours. And we can only see DNA release or NETs release after 3 hours, stimulating the cells with different concentrations of RSV. And we pre-treated the cells with reactive oxygen species the generation inhibitor, actually NADPH oxidase enzyme inhibitor, DPI. And when we pre-treated the cells with DPI, we could block NET release induced by RSV. So characterizing that RSV induces the classical ROS-dependent netosis, which is the most well-studied mode of netosis. And we can see here that RSV also induces and is trapped on these neutrophil extracellular traps, that we can see RSV fusion protein here trapped in these DNA fibers, and RSV also induces NETs release coated with neutrophil elastase and myeloperoxidase. So neutrophils, RSV is able to induce neutrophil extracellular traps, but the virus is trapped in these DNA fibers. However, netosis and necroptosis can be interconnected. So recent evidence suggests that necroptosis proteins participate in NETs release. So once neutrophils are stimulated, they generate large amounts of ROS. ROS will then activate and phosphorylate RIPK1, RIPK3, and MLKL. And once these 3 proteins are activated, they will form a platform called the necrosome, and necrosome activation will lead to plasma membrane rupture and consequently the release of neutrophil extracellular traps. So we wanted to check if RSV would induce netosis dependently on the proteins of, on the necroptosis proteins. So we pre-treated human neutrophils with RIPK3 inhibitor or MLKL inhibitor. And when we blocked both RIPK3 and MLKL, neutrophil extracellular trap release is completely blocked. We then measured LDH release in these human neutrophils as a marker of plasma membrane rupture, and we can see that increasing concentrations of RSV are able to induce LDH release from human neutrophils, indicating that neutrophil plasma membrane is ruptured. And when we block MLKL, LDH release is also blocked, indicating that MLKL is necessary for neutrophil lysis during RSV-induced netosis. So here I showed you that RSV is able to infect human neutrophils and leads to NADPH oxidase activation, which will generate reactive oxygen species, but also activates, I didn't show you here, but we saw RIPK1 activation, RIPK3, and MLKL. And necrosome activation will lead to plasma membrane rupture or cell lysis, leading to release of neutrophil extracellular traps. And these traps are coated with neutrophil elastase and myeloperoxidase. We then wanted to understand the role of RSV in inducing a different type of cell death called necroptosis. So many papers have been published showing that several viruses, including some respiratory viruses such as influenza, These viruses can trigger necroptosis in different cell types. And while some papers show that necroptosis promoted by respiratory viruses like influenza is detrimental to the host and responsible for the high morbidity and mortality of infected animals, some studies show that necroptosis protects against influenza virus infection. So necroptosis can be double-edged sword. Depending on the virus or the cell model, it can be detrimental, but it can also be protective. And this paper was published on the Blue Journal, showing that RSV infection promotes necroptosis of airway epithelial cells. But because alveolar macrophages are responsible for orchestrating the immune response in the airways, we were interested to understand if RSV would be able to induce alveolar macrophage necroptosis and what would be the outcome to the host. So we first collected mouse alveolar macrophages through bronchoalveolar lavage, and we infected mouse alveolar macrophages in vitro with RSV or with UV inactivated RSV. And then we measured cell death using a viability dye and this viability dye permeates the plasma membrane of damaged cells. And then we measured percent of cell death using flow cytometry. And we can see here that RSV is able to kill mouse alveolar macrophages, but this effect is dependent on virus replication, since UV inactivated RSV is no longer able to kill alveolar macrophages. And we can also see here that RSV is able to replicate inside these alveolar macrophages, and we are able to measure RSV viral titers in these cells. And we also collected 2 different clinical isolates from infants hospitalized for RSV bronchiolitis, and we infected human monocytes with these clinical isolates, and we can see that both are able to kill human monocytes. So not only the lab-adapted strain of RSV is able to kill macrophages, but also clinical isolates are able to kill human monocytes. So we then measured cell death in different human monocytic cells. So we can see here that the lab-adapted strain of RSV is able to kill human monocytes, THP1 cells, and U937 cells differentiated to macrophage-like cells using PMA. And RSV is also able to replicate inside all of these human monocytic cells. Here we have human monocytes, THP1 and U937 cells, and we used as a positive control for RSV replication A549 cells, since these cells are highly susceptible to RSV infection. So RSV is able to infect and replicate in all of these different human monocytic cells. So a critical step for necroptosis to occur is caspase-8 inactivation. So depending on the cell death stimulus, for example, we have here TNF. TNF will induce, if caspase-8 is active, TNF will induce apoptosis. The cell will die by apoptosis. But if caspase-8 is inactivated, for example, using caspase-8 inhibitor, pharmacological inhibitor of caspase-8, the cell death switches from apoptosis to necroptosis. So because most of the papers published on necroptosis will stimulate the cells with a cell death stimulus in the presence of a caspase-8 inhibitor, we wanted to understand if blocking caspase-8 would further induce death induced by RSV. So we pre-treated the cells using ZVAD, which is pan-caspase inhibitor, but in our setting, inhibiting caspase-8 did not further increase cell death induced by RSV. So these results suggest that RSV is sufficient to induce necroptosis. So RSV does not require caspase-8 blockade to induce necroptosis, at least in macrophages. So we then wanted to check the role of necroptosis proteins during RSV infection in macrophages. So we collected alveolar macrophages from RIPK3 knockout mice or wild-type mice, and we infected these alveolar macrophages in vitro. And we can see here that RIPK3 knockout alveolar macrophages are completely protected from RSV-induced death. And similarly, when we pre-treated human monocytes with necrosulfonamide, which is MLKL inhibitor, human monocytes are almost completely protected from RSV-induced cell death. And we can also see that RSV induces RIPK3 activation after only 6 hours of infection, and also RSV induces MLKL activation. So RSV induces macrophage necroptosis through RIPK3 and MLKL activation. And we tested the role of necroptosis proteins in virus clearance. So we pre-treated human monocytes with RIPK1, RIPK3, and MLKL inhibitors. And we can see that blocking necroptosis proteins induce a significant decrease in viral load in human monocytes. And when we collect RIPK3 knockout macrophages from these mice, these cells have a significantly decreased RSV viral load, indicating that necroptosis proteins are detrimental to virus clearance. So somehow these proteins promote RSV replication in alveolar macrophages and human monocytes. We then infected RIPK3 knockout mice with RSV, and we can see here that RIPK3 knockout mice are completely protected from RSV-induced weight loss compared to wild-type mice. These mice also exhibit a significantly lower viral load compared to wild-type mice. And they also present decreased peribronchial inflammation score, but not perivascular inflammation score. And RIPK3 knockout mice present a significantly lower frequency of alveolar macrophage of necrotic alveolar macrophage in their bronchoalveolar lavage. So these data indicate that RIPK3 exacerbates lung pathology during RSV infection, and this is partially coupled to alveolar macrophage death. So because TNF is the most well studied necroptotic trigger, we wanted to understand the role of TNF during in vivo RSV infection. And we infected TNF receptor 1 knockout mice or wild type mice. And we can see that TNF receptor knockout mice are protected from RSV-induced weight loss. These mice also present a significantly lower viral load in their lung tissue, and they exhibit significantly lower peribronchial and perivascular inflammation score. These mice also presented significantly lower RIPK3 expression and MLKL expression in their lung tissue compared to wild type mice. And they also present a decrease in the frequency of necrotic alveolar macrophages in their bronchoalveolar lavage. So altogether, this data suggests that TNF receptor 1 signaling mediates lung pathology and alveolar macrophage necroptosis triggered by RSV infection. And finally, we wanted to understand the contribution of TNF during RSV infection. So we collected nasal wash from infants hospitalized with RSV bronchiolitis, or infants hospitalized with infected with other respiratory viruses. So we can see that those infants hospitalized for RSV bronchiolitis present significantly higher levels of TNF in their nasal washes compared to those infected with other respiratory viruses. We then wanted to understand the contribution of TNF in disease severity. So we performed a correlation between levels of TNF and the need of supplemental oxygen therapy, and we couldn't find a significant correlation between the levels of TNF and the need of oxygen, but we did find a positive correlation between the levels of TNF and the length of hospital stay. And we can see here that the infants who stayed more than 7 days in the hospital presented higher levels of TNF in their nasal washes compared to those who stayed less than 7 days in the hospital. So these data suggest that elevated levels of TNF in the nasal washes positively correlate with disease severity in infants hospitalized with RSV bronchiolitis. So for this part of the work, we were able to show that RSV infect alveolar macrophages and also human monocytes, and actively replicates inside the cells. Viral replication will lead to TNF release, and TNF will activate RIPK1, RIPK3, and MLKL, leading to necroptosis. Massive induction of necroptosis will translate to weight loss, increased viral load, and increased lung inflammation in mice infected with RSV. So a part of the work that we do in the lab with cell death, with inflammatory modes of cell death, I wanted to share with you some of the work we have been doing to find potential therapeutic targets and potential antiviral treatments for RSV. So we have been studying BRAF kinase as a potential therapeutic target for RSV infection. So the RAF family of serine protein kinases mediates mitogen activated protein kinase or MAP kinase signaling. So BRAF leads to the activation of MEK and ERK, and these proteins are involved in the regulation of different critical events in cells, including cell proliferation, cell survival, cell migration, and differentiation. So RSV has been shown to promote RAF kinase activation, but the role of RAF kinase during infection is not well understood. So we know that RSV is an FDA-approved small molecule kinase inhibitor that targets BRAF. Dabrafenib is actually an anti-cancer drug used to treat metastatic melanoma. So we wanted to understand if BRAF kinase inhibition or blockade will protect airway epithelial cells from RSV infection. So this work has been led by my PhD student, Julia, and we first infected A549 cells with increasing concentrations of dabrafenib, and we can see in the presence of RSV infection, and we can see that dabrafenib dose-dependently impairs RSV infection of A549 cells, and only the highest concentrations of dabrafenib are cytotoxic to A549 cells. And because dabrafenib is an anti-cancer drug, we measured cell numbers after RSV infection and after RSV infection and in the presence of dabrafenib. And we know that after 48 hours of infection, which is the peak of infection, A549 cells die because they are highly susceptible to RSV infection. So we see a decrease in cell numbers, but when these cells are treated with dabrafenib, there's we can't see a further decrease in cell numbers. So there is no difference in cell numbers between RSV infection and dabrafenib treatment. And in fact, dabrafenib protects A549 cells from RSV-induced death as measured by LDH release as a marker of plasma membrane rupture. So in the lab, we use a recombinant strain of RSV, which express GFP. So every time the virus replicates inside the cells, GFP is expressed and the cells turn green. So we can see here that both the prophylactic and the therapeutic treatment with dabrafenib protects the cells from RSV infection. So for the prophylactic treatment, we treated the cells 24 hours before infection, and for the therapeutic, we treated the cells 24 hours after infection. And we can see that both prophylactic and therapeutic treatments induce a significant decrease in GFP expression and also a significant decrease in infection rate. We also wanted to study a more physiological cell model which is a non-tumor cell model. So we infected immortalized human bronchial epithelial cells, the HBAC3KT cell line, with RSV, and we treated the cells with the dabrafenib for 24 hours before, after infection, the therapeutic treatment. And we also see that the dabrafenib treatment, the therapeutic treatment, protects human bronchial epithelial cells from RSV infection, leading to significant decrease in RSV viral loads in these cells. And in collaboration with Dr. Theo Moraes at Sick Kids, we collected nasal epithelial cells from healthy adults, and we infected these primary nasal epithelial cells with the dabrafenib for different time points, and we treated the cells with the dabrafenib for 24 hours, and we can see here GFP expression throughout 72 hours post-infection, but we can see a decrease in GFP expression. When we measured RSV mRNA expression in the cells after 72 hours of infection, we see a trend to decrease in RSV mRNA expression levels, but likely because we have only small numbers of these samples, our experiment did not reach significant statistical significance, so we likely will need to increase the samples for these experiments. So we wanted to understand if RSV and the dabrafenib would induce a change in A549 cell protein. So we performed proteomic analysis with collaboration with my colleague, Dr. Kevin Coombs here at the department. And we can see that both RSV and the dabrafenib induce significant changes in the A549 cell proteomes. So we can see here in the right-hand corner that both RSV and RSV in the presence of the dabrafenib induce changes in some antiviral proteins such as IFIT1, ISG15, and IFIT3. So we can see that RSV and the dabrafenib induced changes in many different antiviral proteins, but the ones that changed the most were IFIT3, ISG15, and IFIT1. And the dabrafenib was able to decrease the expression of these antiviral proteins. When we measured inflammatory pathways, we can see that the dabrafenib induced a decrease in MUC5B protein, which is a protein involved in mucus secretion. However, the dabrafenib induced an upregulation in MUC13, so suggested that there must be or there might be a compensatory mechanism at play here during RSV infection RSV infected cells treated with the dabrafenib. And when we looked at cell death pathways, we can see that the dabrafenib induced a downregulation of MLKL, which is a protein involved in cell death. Then we measured the levels of ISGs, interferon-stimulated gene expression, in A549 cells and HBAC cells. So we measure IFIT1 and ISG15, and we see that for A549 cells, the dabrafenib was not able to regulate the levels of these two ISGs. However, in human bronchial epithelial cells, dabrafenib significantly decreased the levels of IFIT1 and ISG15, likely because dabrafenib significantly decreases RSV viral loads in these cells. We also measured levels of type 1 and type 3 interferon released from A549 cells and HBACs. So for interferon alpha, dabrafenib was not able to reduce interferon alpha levels, but it significantly reduced interferon beta and interferon lambda 1 levels in A549 cells. And for human bronchial epithelial cells, dabrafenib or RSV infection didn't affect interferon alpha levels, but dabrafenib significantly decreased interferon beta and interferon lambda 1 levels compared to RSV infection alone. So dabrafenib is able to differentially modulate levels of type 1 and type 3 interferon from A549 cells and human bronchial epithelial cells. So for this part of the work, I just showed you that targeting BRAF kinase using dabrafenib protects A549 cells and human bronchial and nasal epithelial cells from RSV infection. Dabrafenib modulates the A549 cell proteome, regulating the expression of proteins involved in antiviral and inflammatory responses and cell death pathways. Dabrafenib also differentially regulates RSV-induced ISG expression in A549 and human bronchial epithelial cells, and it also differentially regulates RSV-induced type 1 and 3 interferon release from A549s and human bronchial epithelial cells. So I'd like to thank all of my students in the lab, my collaborators in Brazil who were critical for our work with necroptosis, and my collaborators here at the University of Manitoba and at SickKids, and the financial support. Thank you. So I will stop sharing my screen. I'm happy to answer any questions.
Thank you so much for that. That was very insightful. And I would first open the questions. Any trainees want to ask any questions, please? And faculty members for any questions for Dr. Porto? I don't see anything in the chat either. We have about 30 people in the current call. So Barbara, let me just start with the questions. So have you looked at pathways of pyroptosis in this?
That's what we are doing now. Yeah.
Oh, did I hit? Okay, I hit it.
we are studying pyroptosis induced by RSV in airy epithelial cells, not in macrophages.
Okay, and the induction of pyroptosis, how are you going about it? Like with RSV, so RSV can cause pyroptosis?
Yeah, so we are able to measure IL-1 beta release. We were able to see that RSV induces caspase-1 expression, and when we block caspase-1, we no longer see LDH release, and we are also optimizing actually Western blots to measure Gasdermin D expression. So Western blots are always tough
tricky.
when we start
Yeah.
working with that. So we were able to see the expression of full length Gasdermin D, but when Gasdermin D is cleaved, the portion of the protein responsible to induce membrane pores is the cleaved one, the N-terminal of the protein. So we were able to measure full length, but we are still struggling to measure N-terminal, the cleaved one.
Possibly because it's in the membrane, and membranes are always difficult.
Yeah.
For protein, this thing for the Western blot. Yeah, I'm aware of that. I have Nadia Milad, who is a PhD student at the Hirota lab, say that it was an amazing talk, and asks a very important question. Do you think this approach could be applicable to other viral infections, and what about co-infections with bacteria?
Hi, Nadia. Thank you for your question. So you mean the approach you mean for BRAF kinase inhibition or necroptosis? So
Nadia, you can come on. You can unmute and ask the question to Dr. Porto yourself.
Okay. Yeah. Thank you.
Right. Thanks.
Great talk again.
Thank you.
Yeah. No, I was talking more about your treatment, the drug in question. Like if it's working really well for RSV, there are so many other viruses that we have very few effective treatments for. So I just wondered if you tested any.
Yeah. No, we. For now, we only tested the brafenib for RSV infection, but there's one paper, but not a complete one, showing that the brafenib protects viral cells against SARS-CoV-2. And there is another paper showing that the brafenib protects cells from Zika virus infection. But
oh, cool.
Yeah.
And those were different cell types too. So it's not just epithelial cells.
No, these are different cell types. Yeah.
Very cool. Thank you again.
Yeah. Thank you.
That's really cool. Good question, Nadia. That makes a lot of sense. So, which means the way SARS-CoV-2 can induce NETosis, which has been shown, is possibly through the BRAF pathway. Then
maybe, yeah. I haven't seen a paper correlating BRAF activation and NETs release, but maybe, yeah.
But indirectly, if the brafenib is working, that means it's working by inhibiting the BRAF pathway, right? Or am I getting it wrong?
Yeah. But we haven't tested the brafenib in neutrophils, only in airy epithelial cells. Yeah.
I see.
Yeah.
Okay. So you think in neutrophils the pathway is more towards RIP kinase 3?
Well, we showed actually that RSV induces NET release through MAP kinase activation, but we only measured P38 and ERK, which are downstream of BRAF. But yeah, it's probably, it's very possible.
No. Well, MAP kinase makes more sense, actually. That's what we see as well. Okay. Any other questions from anybody else? Come on, don't be shy. This would have been wonderful if we could have had you here, Barbara, but I think you're coming to McMaster anyway soon, right?
No, not yet.
No. It's Diana. It's Diana.
Diana is coming.
But hopefully we'll try and get you in for an in-person as well. But this was so good. If there are no more questions from our students, our trainees and faculty who are online right now, I can see Alex ask a question. Again, Alex is a PhD student, if I'm not mistaken. Thanks for the great talk. Have you seen any data with primary human epithelial cells? I wonder if there is a specific cell that would be affected by dabrafenib, like AT1 or versus AT2. Alex, you can come online if you want to have a chat, direct conversation with Dr. Porto.
Okay, I will start asking, answering Alex's question. So, Alex, for the brafenib, we tested human nasal epithelial cells, and we see a reduction in RSV-GFP expression and also in RSV mRNA levels. But because we have only tested so far 5 patients, we were not able to reach statistical significance, but we didn't test like in specific different types of epithelial cells, like 81 or 82, because these cells, we culture them all together. So it's a mix of all of the different cell types. And we have goblet cells, we have all of the cells together. So it's actually very nice, because when we work with these cells, they actually produce mucus. So we can see like the cilia beating and the mucus flowing. It's really cool.
So you get the nasal epithelial cells and put them in culture and make ALIs out of it, and then you infect.
We culture the cells in an ALI culture, air liquid interface.
Cool. If I remember the graph that you showed, and it showed MUC13 to be restored, elevated when you add dabrafenib. So MUC13 is more of a receptor for mucins that's expressed on epithelium, and MUC5B is also restored, because MUC5B is the constitutive.
MUC5B is downregulated and MUC13 is upregulated.
No, but when with dabrafenib, it reversed.
Dabrafenib induces a decrease in MUC5B, but upregulates MUC13. It has like opposite effects.
Okay.
So so that's why we think that there might be a compensatory mechanism, you know.
Well, MUC5B is a constitutive good mucus, so it's not a bad mucus. So your epithelium should be making that mucus anyway, which is a good thing.
Okay.
Anyway, any other questions? Anybody? If not, this was wonderful. Thank you, Barbara, for coming in and sharing your data and research with us. It's always nice to hear your talk. Thank you so much, and hopefully we'll see each other soon.
Yeah, hopefully. Thank you so much for inviting me, Manali. It was a pleasure.
It's a pleasure.
Thank you, everyone.
Bye.
Bye.